let's just jump right into this because we are starting with what is honestly the ultimate paradox of classical biology. For decades, the common wisdom, as Yale scientist Nicole Malvin Carr points out here, was that the biological world is just too warm, too wet, and honestly, way too noisy to sustain fragile quantum effects. Subatomic weirdness like quantum entanglement. That was supposed to exist only in these highly isolated, ultra-cold lab vacuums, definitely not inside the messy, boiling soup of a living cell. But, as we're about to see, the biological world holds a massive surprise. Welcome to this explainer. Today, we're taking a deep dive into a completely paradigm-shifting concept, quantum entanglement in enzymes. We're going to explore exactly how entanglement facilitates biological functions, how quantum bonds actually operate, and the real physics behind molecular recognition. We aren't just talking about abstract chalkboard math here. We're talking about how the very enzymes keeping you alive right this second might actually be functioning as biological quantum computers. So here is our flight plan for today. Number 1, the biological quantum mystery. Number 2, the physics of bonds. Number 3, entanglement in enzymes. Number 4, the quantum evolutionary origin. And finally, number 5, nutritional quantum biology. Section 1, the biological quantum mystery, the noisy biological environment versus subatomic reality. To really wrap our heads around this, we have to completely reframe how we view the cell. It's not just a bag of interacting chemicals playing a classical game of microscopic billiards. It's a highly advanced quantum-capable environment. Now, what's incredibly interesting here is how we define quantum entanglement in a strictly biological context. We're not talking about sci-fi teleportation, right? We're talking about the sharing of quantum correlations between bonded biological systems. Through phenomena like electron or proton tunneling, particles share states in ways that absolutely bypass the classical rules of thermodynamics. It's a tangible, physiological reality happening inside your body, allowing biomolecules to communicate with mind-bending efficiency. Section 2, the physics of bonds, finding the culprit at the subatomic level. To find out where this quantum resource actually lives, we need to zoom all the way in on the periodic table. We're specifically focusing on the interactions between elements like hydrogen and carbon. When you compare these bonds, a standard, pure covalent bond involves shared electrons that are identical and entirely indistinguishable. The source research reveals that from a purely quantum information standpoint, a pure covalent bond is literally useless for storing entanglement. There's just no usable information there. But a hydrogen bond, that is a completely different story. Because it has a partial ionic character, meaning there's an unequal sharing of charge, it turns into a quantum goldmine. It is rich in entanglement generated by particles tunneling back and forth. Now, early attempts to model this mathematically hit a massive wall with what researchers call the flawed 2-qubit model. It tries to treat two identical electrons as separate, distinguishable qubits of information. But as we just learned, you cannot tell identical electrons apart in a bond. The math here looks beautiful, for sure, but as a biological model for entanglement, it fundamentally fails. Why? Because a simple mathematical sign flip could arbitrarily change the entanglement from 0 to maximum without changing anything about the bond's physical reality. But then, a major breakthrough elegantly resolved this. Instead of tracking individual, identical electrons, physicists shifted to a one-quitrit representation. So instead of a qubit, which has 2 states, a quitrit has 3. They modeled the atomic orbital itself, asking, does this specific orbital hold 0, 1, or 2 electrons? This totally solves the indistinguishability paradox. It mathematically proves that electron tunneling creates highly useful quantum entanglement as long as that partial ionic character exists in the bond. This is exactly how quantum bonds operate in biology. And that brings us to the absolute climax of the physics. When a proton acceptor atom is introduced to a hydrogen bond, the proton itself, the literal nucleus of the hydrogen atom, can delocalize. It essentially smears out, existing in a strange state of probability right between the two atoms. This back-and-forth proton or electron delocalization creates the ultimate biological quantum state. This is not just theory, this is the fundamental, underlying physics of how molecules bind together in your cells. Section 3. Entanglement in enzymes, how biology utilizes quantum resources. Now let's scale up from those tiny raw bonds and see how massive protein structures actively use this. Specifically, we're looking at enzymes. Forget that old high school biology analogy of a lock and key where physical shapes just randomly bump into each other until they fit. If an enzyme leverages quantum entanglement through its hydrogen bonds, it executes what's basically a quantum handshake. The math shows an enzyme can perfectly discriminate between exactly 3 to the power of n-1 specific target molecules, or ligands, using just n hydrogen bonds. The physics behind this recognition is just staggering. It's literally synchronizing across multi-dimensional space to find its perfect molecular match. The researchers outline this as an actionable entanglement swapping protocol. You can think of it as a futuristic computation happening natively inside your cells. Step 1. The enzyme effectively measures the quantum state of the incoming ligand. Step 2. It performs local quantum operations on its own internal hydrogen bonds. Step 3. If the entanglement perfectly matches, it triggers the target state, a physical conformational change in the enzyme that causes a biological reaction. This right here is the precise mechanism of how entanglement facilitates biological processes. Section 4. The quantum evolutionary origin. Evolution driven by quantum mechanics. Now that we see how enzymes function as quantum processors today, let's shift into the deep past. How did life itself evolve to act like a quantum computer? Why did quantum DNA evolve in the first place? Back in 1944, physicist Erwin Schrödinger first postulated that quantum mechanics had to underpin biological order, but modern analysis of the pre-DNA world takes this so much further. Think about ancient primordial enzymes, synthetases. They had a massive problem. They were structurally incredibly small, but they still had to accurately sort through a huge variety of molecular ligands to build the very first building blocks of life. Because they completely lacked complex physical structures, the physics strongly suggest they leaned heavily on quantum entanglement. They used fewer physical bonds to process way more information. This is the origin story of enzymes, laying the quantum groundwork for the highly complex DNA and RNA machinery we have today. Section 5. Nutritional quantum biology, scaling up to the whole holobiont. Finally, let's zoom out from ancient evolutionary history to the macro scale of modern human health. We are stepping into the cutting-edge field of nutritional quantum biology to see how the science behind entanglement ties into your actual environment and diet. This map bridges the gap between abstract physics and your bodily health. Notice how quantum coherence and electron transfer via light are directly linked to your bioenergetics. Notice how quantum tunneling isn't just a neat enzyme trick, but is intrinsically tied to epigenetics. And electromagnetic fields governed by quantum entanglement have profound applications in inflammation and immunity modulation. We are, in essence, a unified ecological network, a holobiont, entirely governed by quantum rules. To summarize the profound implications of all this, researchers propose that quantum DNA repair is continuously operative across your entire lifespan. There is even the mind-bending possibility of intergenerational epigenetic entanglement, meaning the quantum impacts of an environment might actually be passed down to your kids. Your mitochondria, the famous powerhouses of the cell, are actively acting as quantum transducers, deploying electron and proton tunneling just to create energy. On top of that, high-spin iron ions in your electron transport chain generate tiny magnetic fields that protect your cells from reactive oxygen species. It is an absolute masterclass in subatomic engineering, which leaves us with this final, genuinely awe-inspiring thought to ponder. We started out by questioning how the messy, noisy biological world could possibly host delicate quantum states. Yet, the data absolutely reveals that you are, right now, a walking, breathing quantum architecture. If our enzymes are actively operating like quantum computers to keep our cells functioning, what other profound mysteries does our DNA still hide? Thank you so much for joining me for this explainer. I highly recommend diving into the source materials if you want to explore the subatomic machinery of life even further.